Last video lecture for Chapter 3, and then we'll move on over to Chapter 4. We're going to talk a little bit about the Archaea, the other big group of prokaryotes, and then we'll also talk a little bit about classification as well. All right, so classification. So I'm not real big on students who are going into the health sciences and not planning a career in biology or microbiology, learning a lot about the classification of microorganisms. So we are just going to hit some highlights here. Again, these are ways of dividing up living things into categories based on their evolutionary relationships with each other, how closely related they are to each other. And if you guys remember from back in Chapter 1, we talked about how on the Earth you got all living things, and then they're divided up into 3 domains. Okay, so one of those domains is Eukaryota, and then one is bacteria. So Eukaryota, those are all the eukaryotic living things, obviously, and that includes everything that you're familiar with the naked eye, animals and plants and little protozoans, some of the fungi, helminths, the parasitic worms, and so forth. All of those things fall into the eukaryotic category. Then we have bacteria, which we've been talking extensively about here in Chapter 3. And then finally, you have a third major group called the Archaea. So all 3 of those are domains of living things. So every living thing on the planet, and remember the viruses are not included, are divided up into one of these 3 groups. And so everybody within a particular domain, all of the Archaea, are more closely related to each other than they are to the bacteria. And all of the bacteria are more closely related to each other than they are to the Eukaryota or the Archaea. All right, and then another, now among your prokaryotes, so again, your, get rid of that, go away. Go away. There we go. All right, your prokaryotes are your bacteria and your Archaea. So they both fall into that prokaryotic cell category, but you have 2 different domains there. Now, one way of classifying your prokaryotes is to divide them into 4 divisions, and 3 of those divisions are bacteria. So these are all in the domain bacteria, and then you have this division, the Mendocicutes, which are, some of these have odd pronunciations. That represents your Archaea. So for a long time, people thought Archaea were just bacteria. So some of this classification is older, and then later it was appreciated that the Archaea were so different down at the molecule level from bacteria that they represented a completely separate domain of living things. You have your Gracilicutes, those are bacteria with gram-negative cell walls. Firmicutes, those are bacteria with gram-positive cell walls. And then Tenoricutes are bacteria with no cell walls. So if you remember earlier in Chapter 3, we talked about those 3 categories, the characteristics of bacteria, that how they could be divided up based on their cell wall structure. So there are names for bacteria that fall into those categories. And then finally, the Archaea can also be referred to as the Mendocicutes. All right, but let's talk a little bit more about the Archaea. So again, they are their own domain of living things. Like bacteria, they're single-celled or unicellular. Similar to bacteria in overall structure, so if you looked at them under a microscope, you wouldn't be able to tell just based on microscopic appearance whether an organism was an Archaean or a type of bacteria. It really gets down to the detailed molecular level. So the cell wall structure, for example, differs. So Archaea do not have a peptidoglycan cell wall. Their cell walls are built from other chemical components. And interestingly, the cell membranes, so we talked about how cell membranes are built from phospholipids. That's true for bacteria, and it's also true for eukaryotic cells, but the Archaeans build their cell walls from special types of lipids. All right, and so really because of those two of those key things there, the different cell wall structure and these different cell membrane lipids that are used, that lets us know these archaea are often their own category. They're not all that closely related on the evolutionary family tree to the bacteria and the eukaryotes. All right, the archaea, one thing that kind of distinguishes them from the bacteria is that many or most of them are adapted to extreme environments. And I kind of hesitate to use words like all of them because that doesn't generally apply very well in biology, and especially microbiology where you have so much diversity. And so I'm a little hesitant to even use the word many, because who knows, we might discover that there are archaea in pretty normal environments more and more as we continue to look in the future. But so a lot of the first archaea to be discovered, though, were found to live in bizarre conditions, very high temperatures, low pH, high pH, acidic alkaline type conditions, environments with a lot of sulfur gases around, so environments that would kill us and would be hostile to many of the microorganisms that we commonly work with in the laboratory and the ones that really impact our daily lives. Quite a few of these archaea are adapted to these extreme conditions. So if you've ever been, this is a picture from Yellowstone National Park, so if you've ever seen the really colorful pools of hot steaming water there, a lot of that color is coming from archaeans that live in the very hot water environments of those springs out in Yellowstone National Park. All right, I do want to go over some terminology here because these words get thrown around a lot. And one of them is species. So we've already talked about genus and species. And so for animals like us, it's pretty obvious that we're a different species from chimpanzees and gorillas and things like that. But when you're a little bacterial cell, it becomes a little more difficult to define what a species is, because so many of them look alike, they're all microscopic, and it gets down to a detailed level, and you guys will see this as you move further along in the virtual laboratory exercises later on in the semester, and you start to take advantage of these different chemical characteristics and so forth that bacteria have. You can take advantage of that to help you identify an organism. So when we're thinking about traits and characteristics of microorganisms, a lot of times it gets down to a chemical level. Does this bacterial cell feed off of this kind of sugar? Is it able to make this type of compound? So a little bit more detailed than just observing, making observations with the naked eye. So a species of bacteria, generally, if you got two bacterial cells and they have a very similar pattern of traits, they're likely in the same species. And within a species, generally the members of that species, 70 to 80% of their genes will be shared with each other. So they've got to have a pretty high level of genetic identity, but not 100%. Because within a species, you can have variations that do occur. So you can have a species like Escherichia coli or Escherichia coli, good old E. coli. And then within that, you can have smaller groups. So all the members of that species are closely related enough to be considered to be a species. But within that species, you can have smaller groups that have shared characteristics that are distinct from the other members. And there is some terminology associated with that, like strain. And I do want to point out, a lot of students, and I think it's because when we start studying DNA, more so later in the semester, we start talking about strands of DNA, but a lot of students will talk about strands of bacteria, strands of viruses, and it's not strand, it's strain. There are different strains of E. coli, for example. So you've no doubt have heard about people getting sick from E. coli on the news. Well, most E. coli doesn't make you sick. It lives in your colon and keeps you happy and healthy as long as it stays in the colon and doesn't go somewhere in the body where it's not supposed to be. But there are strains of E. coli that produce toxins, toxic substances that are able to cause you to become sick. So they cause inflammation of your colon, kind of like we were talking about with C. diff. And so those are special traits or characteristics that they have that the normal members of the E. coli species don't have. So that sets them apart as a separate strain. Okay, and then some other terminology kind of in the same vein. Subspecies. So some species can be divided up into subspecies groups, kind of similar to the concept of a strain. Same thing with types. You might hear type A, type B, type this, type that. Or serotypes. Now, sero sounds like serum, which reminds us of the blood. And it's more specifically here referring to the immune system. How does your immune system react to the members of a species of microorganisms? And so different serotypes, really what that means is that there are some differences on they're all members of the same species, but on their surface is a particular serotype. The molecular patterns there differ from another serotype, which differ from another serotype. And since that's what your immune system is looking at a lot of times, it's the surface of the microorganism. If a particular serotype has altered its surface so that it's a little bit different from another serotype, then your immune system may remember that you've been exposed to serotype number 1, but not serotype number 2. So it kind of gets down into that level, your immunity that you develop to these microorganisms after you get exposed to them. So we'll hear that terminology during the semester as well. Some of this applies to viruses. I know viruses are not living things, so we don't have a genus and a species name for a virus because they don't fall into the broad classification of life forms here on the planet. But but you will hear some of that terminology used with viruses. There's a new strain of influenza, like the H1N1 strain, or there's a new serotype of the common cold virus. You will hear some of that terminology used clinically. All right, so that actually concludes Chapter 3. We're going to move on to Chapter 4 with the next video lecture. Please read the instructions in your study guide carefully for Chapter 4 because we will not be covering the whole chapter. We're just going to hit some highlights from that chapter as we finish up this first part of the course.